Today we're talking about two closely related but clinically very different genera, Campylobacter and Helicobacter. Both are curved, gram-negative, microaerophilic bacteria. Both colonize mucosal surfaces. Both have zoonotic potential, but they cause very different diseases across species, from diarrhea in puppies and calves, to abortion in sheep, to gastric disease in dogs and cats. By the end of this session, you'll have a clear mental map of which species matter, what diseases they cause, and how we diagnose and manage them. Let's get to it. Campylobacter and Helicobacter share several key features. They're curved or spiral gram-negative rods, and they thrive in low oxygen environments, especially mucosal surfaces like the GI tract or reproductive tract. They're also fastidious, meaning they're tricky to culture and often require special media or atmospheric conditions. And importantly, several species are zoonotic, making them relevant to both veterinary and public health. These are the major Campylobacter species you'll encounter. Campylobacter jejuni is the classic cause of diarrhea in dogs, cats, and humans, and is strongly associated with poultry. Campylobacter coli is common in pigs and also zoonotic. Campylobacter fetus venerealis causes bovine genital campylobacteriosis, a reproductive disease. Campylobacter fetus causes abortion in sheep and goats. Each species has its own niche and disease pattern. In small animals, C. jejuni causes diarrhea, especially in young, stressed, or crowded puppies and kittens. Clinical signs range from mild loose stool to watery diarrhea with mucus. Many infections are self-limiting, but zoonotic transmission is possible, especially in households with children or immunocompromised individuals. Poultry is the major reservoir globally, which is why food safety messaging emphasizes proper handling and cooking of chicken. In livestock, Campylobacter species cause a range of diseases. Calves may develop diarrhea from C. jejuni. Pigs commonly carry C. coli, which can cause enteritis. Sheep and goats experience abortion storms from C. fetus. Cattle can develop infertility and early embryonic loss from C. fetus venerealis, which colonizes the reproductive tract and spreads during breeding. Enteric Campylobacter species colonize the intestinal mucosa and invade epithelial cells. This invasion triggers inflammation, leading to diarrhea that may contain mucus or blood. In most animals, disease is mild, but in younger immunocompromised hosts, it can be more severe. Reproductive Campylobacter species behave very differently. C. fetus venerealis spreads during natural breeding and causes early embryonic death in cattle. Cows return to estrus irregularly, which is a classic sign. C. fetus causes abortion in sheep and goats, often late term. These outbreaks can be dramatic and economically significant. PCR is the most sensitive and practical diagnostic tool. Culture is possible, but requires special atmospheric conditions and selective media. For abortion cases, placenta and fetal stomach contents are the best examples. For enteric disease, fecal samples are appropriate, but remember that Campylobacter can be part of normal flora, so interpretation requires context. Helicobacter species are best known for H. pylori in humans, but animals have their own species. Dogs and cats commonly carry H. felis and H. hellmanni. Pigs carry H. suis, which may contribute to gastric ulceration. Horses have H. equorum, though its clinical significance is still being studied. In dogs and cats, Helicobacter species are extremely common. Many animals carry them without any clinical signs. When disease does occur, it usually presents as chronic vomiting, intermittent inappetence, or signs of gastritis. Histopathology often shows lymphocytic or plasmacytic inflammation, but because Helicobacter can be found in healthy animals, diagnosis requires correlation with clinical signs. In pigs, H. suis is associated with gastric ulceration, especially in the pars esophageal, stress, feed changes, and management factors also contribute. In horses, H. equorum is frequently detected, but its role in disease is still unclear. Helicobacter species colonize the stomach by producing urease, which breaks down urea into ammonia and CO2. The ammonia neutralizes stomach acid, creating a microenvironment where the bacteria can survive. Chronic inflammation develops over time, which can lead to gastritis or ulceration. Diagnosis requires gastric biopsy, either via endoscopy or at necropsy. Histopathology can show organisms in the mucus layer or gastric pits. PCR confirms species. Rapid urease tests can support diagnosis but are not species specific. Treatment mirrors human protocols, a combination of antibiotics plus acid suppression. Common regimens include amoxicillin, metronidazole, and a proton pump inhibitor. Because many animals carry Helicobacter without disease, we only treat symptomatic animals with confirmed gastric inflammation. To wrap up, Campylobacter and Helicobacter are curved microaerophilic bacteria that cause very different diseases across species. Campylobacter is a major cause of diarrhea, abortion, and infertility. Helicobacter is primarily a gastric pathogen. PCR and targeted sampling are essential for diagnosis, and treatment depends on the species and clinical presentation. If you found this helpful, please like and subscribe. Thanks, and I'll see you next time.